So this is our last video for the last topic of biology. So this is the kidney. Well, not the last video, it will take 2 videos to cover all the material. Charlotte's doing this because she knows I'm super sad because it's the last topic, and I really wanted to teach you this in college, but it'll still be epic. Okay, so I hope you enjoy it. Okay, so the first thing that I want you to do is to think about some key terms that we've already met in the course and tell me what you know about them. So can you write them down on paper in front of you? Diffusion, active transport, water potential, osmosis, microvilli and mitochondria. We're going to meet all these key terms in the kidney one last time. So everything you can, so pause me now, write everything down that you know about each of those. Okay, so you should have had some time to think about each one and put down what you remember. Here are the key things that I would expect. For diffusion, I'd expect you to have some sort of definition, movement of molecules from a high to low concentration. Passive process, doesn't require ATP, and simple diffusion occurs through the phospholipid bilayer. Facilitated diffusion via carrier proteins or channel proteins. For active transport, we're looking for a bit more in your definition. So the movement of molecules from a low to a high concentration, that it requires ATP and a carrier protein. Water potential, it is a pressure, so it's measured in kilopascals. The higher the water potential, the more water. The lower, the more negative, the more solute. So if you add more solute, it becomes lower in water potential or more negative. So that leads us to our osmosis definition. So osmosis is the movement of water from a region of high water potential to a region of lower water potential. So that could be from a more negative to a less negative to a more negative water potential. If you've learned it in those terms, that's okay too. So that could be the movement of water from a higher water potential or a less negative water potential to a lower water potential or a more negative water potential. Microvilli, so these are the folds in the membrane which we've met a number of times for different cells and their specializations, which increase the surface area, that should say, for the movement of molecules. So if we think about the times that we've met that before, it was in the epithelial cell, the small intestine. We're going to meet it again in the convoluted tubule cells of the kidney. So a lot of the kidney is about movement of molecules. And finally, mitochondria. So the site of ATP production via Krebs cycle and oxidative phosphorylation is what we should be writing. It is the site of aerobic respiration, but remember glycolysis, which is also part of aerobic respiration, happens in the cytoplasm. So we were a bit more precise by naming the parts that happen just in the mitochondria. Right, so we're ready to go. So you should have your kidney notes handy as well. Okay, so you should have your kidney student notes running alongside the PowerPoint. Okay, so you can see that the kidney topic looks like quite a small topic in the spec, but actually there's quite a lot of structural information that you need to learn first of all. So you need to know the structure of the nephron, and you need to know all about the different regions of the nephron and how they're specialized in their role. And then we can understand how osmoregulation, so that's the control of water potential of the blood, is achieved through the secretion of ADH. Okay, so a bit of background information about the kidney to start off with. So usually I have a little point to ponder here and say, what do these have? What's common about these pictures? So we've got salt, urea and water. Well, the kidney is involved in obviously balancing the amount of ions and water within the body, but also in getting rid of urea, which is poisonous. And if you have a look at urea, you can tell what it's made from. You can see the amino groups from, you'll be familiar, from our proteins, from our amino acids. This is the bit that is deanimated, removed in the liver, and it then needs to be got out of the body. So it does. We do this in the form of urea. Okay, so let's have a look. How does the kidney achieve that balance in terms of water and the movement of ions, and also get rid of all the urea waste product from your body? So this is your kidney. Okay, so we do need to know a little bit about its gross structure, and just give you a bit of background information in terms of the structure and function of the kidneys, just to give you a bit of context. But it's the nephron really that the exam questions are going to be centered around. There's a lot of quite good application knowledge about diseases of the kidney, which talk about bits of the nephron working or being dysfunctional. And then hopefully today we'll get to the point where you can describe how the kidney produces urine. So again, this is all about the nephron and its structure to function relationship. So let's have a think about what you learned at GCSE in terms of getting rid of waste from the body. We know that there are different organs specialising to remove waste products from the body. So your lungs to get rid of carbon dioxide. The skin, we lose water and ions from the skin, so salt, things like sodium ions. The liver is where I said that deanimation process happens, where excess proteins, amino acids, are broken down and the amino groups are removed and produce urea. And that leads me to where you should know, hopefully, your kidneys are, and that they are involved in removal of unwanted substances, urea, excess water and salts. So that's what we want to learn about, is how that all happens. Okay, so we're going to do a bit of a memory recall task now. So it's a bit of a challenge for you. I'm going to show you a picture of the kidney. Okay, I'm going to talk you through the structure. This is just the gross structure to help you orientate yourself when we're learning about the nephron. So the kidney is surrounded by a capsule. Underneath the capsule, the outer layer is called the cortex. So we'll talk about some parts of the nephron being in the cortex of the kidney. And then we've got the medulla, which we will use quite a lot in our descriptions of movements and things happening in the kidney. So it's knowing where that tissue is, so in the inner of the kidney. Then we've got the pelvis, which connects the kidney up to the urethra. Ureter. I've done that wrong, haven't I? Ureter. So the urethra, of course, goes from your bladder. This is the collecting tube, if you like, from the kidneys that transports urine to the bladder. So ureter. Importantly, there is a synoptic link here to the blood supply to the kidney and away from the kidney. So we've got a branch of the renal artery supplying blood to the kidney and a branch of the renal vein taking blood away. So the examiner will like those two labels and want you to be able to make that link to what we did last year. Okay, so this is now where the memory challenge comes. So I would like you to stop the video and draw that kidney. Label it. Okay, so you should be back with me. I'm hoping that you've been busy drawing and what you've produced is something as beautiful as this. Okay, so if we can check your picture against the original, have you got the outer capsule? Have you got beneath that the cortex? Beneath that the medulla, and then joining to the ureter via the pelvis? So got those layers in correctly. Okay, and then have you got your blood supply right? So renal artery supplying blood to the kidney and the renal vein taking blood away. Super. So now you've got your diagram. I'm going to ask you to imagine that we're turning it that way and we're kind of zooming in on the cortex and the medulla of the kidney. And we're going to look at what is the functional unit of the kidney, the nephron. So all of this tissue is made up of many of these structures. They are structures that are sort of bridge across both the cortex and the medulla. So we'll talk about that tissue and we'll try and orientate that on the next diagram. Okay. In fact, I think in your keynotes it does a little bit. So let's swap back into those. So again, these are just for your background knowledge, so you understand what we're talking about when we're in the talking about the function of the nephron. So here's a little sort of picture to show that we've cut a section, if you like, of the kidney through. This is the cortex, this is the medulla. We've cut that out and we're zooming in on this functional unit, which is the nephron. Okay. Okay, so you've got another task now. I'd like you to have a look at that picture, that nephron picture, in your keynotes, the labels. And I've got a slightly different diagram here, and I would like you to identify A, B, C, D, E, F, and G from your diagram. So stop here and have a go. Use your keynotes. Okay, so by now we should have labels on this diagram, hopefully. So we're looking first of all for this structure here, A, okay, which you should have labelled as the Bowman's capsule. You can see that this bunch of capillaries, this network of capillaries, sits within that Bowman's capsule. That's the glomerulus, but we're looking at A, so that is the Bowman's capsule. So that's the first part of the functional unit, which is the nephron. Then from there, we're moving in to this convoluted tubule. And because it's before the loop, this is called the proximal convoluted tubule, sometimes abbreviated to proximal convoluted tubule, PCT even in textbooks, but always use its full name the first time. Okay. So then if we go to C, this is where you've got to be careful. My picture is a little bit different to yours. You can see that the proximal convoluted tubule is actually joining on the right hand side of this picture. So that means that this side here, C, is actually the ascending limb of the loop of Henle, because it's the bit that's going back up. Okay, so D is the apex of the loop of Henle, so the bottom or base of the loop of Henle, and E then is still the loop of Henle, but this is the descending limb. F, you can see, is all convoluted again, but it's after the loop of Henle, so this is the distal convoluted tubule, and that connects up to the collecting duct, which is G. Okay, another memory challenge now. Okay, I've partially deleted some structures in this nephron diagram. I'd like you to keep this paused on the diagram and then try from memory now to add anything that is missing from the diagram. And that includes parts of labels and also parts of the nephron structure itself. Okay, so make sure you pause there. Okay, you should have unpaused now, so I'm going to be revealing my beautiful diagram. So I've done this beautiful version here. Okay, so what was missing then from this diagram? Well, the first thing you can see that I've drawn in is a capillary network, okay, which is your glomerulus, which is this here. You can see that the blood vessel supplying this capillary network is a wider blood vessel than the one that's leaving, which is a narrower lumen. That's a really important feature. Make sure your diagram reflects that. I've tried to. Okay, so there were some missing labels before and after that blood supply. So before was the efferent arteriole, and after was the efferent arteriole. You don't need to know afferent and efferent. They're not bothered about that anymore. What they're concerned with is that you realise that these are both arterioles. So that must tell you something about the blood pressure inside that capillary network after this process. It must still be a high blood pressure because we're still in an arteriole, yeah, a small artery, even though we're coming out of a capillary bed. And the other thing is that the reason for that. So this idea that we've got a wide vessel in and a narrow vessel out, but we'll come back to that in a minute. Okay, so then you can see that in this diagram, the convoluted tubule was not attached to the descending limb at this time. So this was on this side, the descending limb of the loop of Henle. So you had to connect them up and make sure that it was convoluted. So we'll start wiggling back on itself. And then add the proximal convoluted tubule to that label. Then you needed to connect up your ascending limb up to the distal convoluted tubule, which is what you'd have as your label up there. And then that would connect up to this collecting duct. Okay, and that would update your diagrams, give you a full diagram. In your keynotes, of course, you have got the picture that you would have been referring to here. Okay, it's just a renal capsule instead of Bowman's capsule, so you might want to just update that. On the mark schemes, it's Bowman's for AQA. Okay, so it's this idea then that on my diagram I just showed you, I have the blood supply to the glomerulus and from, but then all the rest of the capillaries in the next capillary network are kind of missing off this diagram. That was just for ease of the diagram. Okay, they are there. And when we talk about things moving out of the kidney, they are going into the blood. So that's important to realise that there are capillaries associated with the whole of the nephron, which is why that's quite a good picture in your notes. Okay, so what we need to do now is to look at the function of each part of the nephron so that we can understand how each part is adapted to its function and how at the end of this process we produce urine, which then is collected by the collecting ducts and forms the urine which leaves the kidneys by the ureter. Okay, so we're starting with the Bowman's capsule with a process called ultrafiltration. Okay, so if you look closely at the Bowman's capsule, and this is a microscope image, you can see that you've got this dense capillary network. You see the red blood cells, and we can see clearly then that the vessel supplying is much wider than the vessel taking the blood supply away from this glomerulus, this special capillary network that sits within a Bowman's capsule. You can also see the Bowman's capsule has got sort of pores within its walls. Okay, you don't need to know the name podocyte, but these cells, these podocyte cells, are kind of have extensions, feet that create these pores. So the idea there are pores is incredibly important. So if I take you into your keynotes, you can see there's a number of things that make this a filter. Okay. So the first thing to note is the difference in those vessel diameters. Okay, because the supplying arteriole is wider than the one taking the blood away, this creates a high hydrostatic pressure within the network, capillary network, the glomerulus of the Bowman's capsule. Okay, so we've got a high hydrostatic pressure in these capillaries here. Now we know the capillary walls are permeable. Okay, now we can see from the Bowman's capsule wall that that's got pores as well. There is a basement membrane, this blue line here, between those two that hold them tightly together, and that's got holes and pores in it as well. So some people call this Bowman's capsule a molecular sieve. So we've got blood full of lots of molecules, lots of water obviously making up the blood plasma, but in there we'd have glucose and salts, so sodium ions, potassium ions, chloride ions. In here we'd have glucose, in here we'd have amino acids, so lots of different molecules. Now unless they're very large molecules, they do get forced out of the blood plasma here into the Bowman's capsule, and they create what is called the filtrate. So that's the resulting filtered material, and it's basically anything that's not very big. Okay, so okay, so how is this Bowman's capsule adapted to ultrafiltrate, if you like, the blood? Well, we've got this high hydrostatic pressure created by a wide vessel supplying and a narrow vessel taking the blood away. We've got capillaries that we know are 1 cell thick, okay, so they're already permeable. We know that they're closely associated, the capillary and the Bowman's capsule wall, by this blue line in the diagram. The basement membrane holds them together. And we know that there are pores in the basement membrane and between the feet of the podocytes, so in other words, in the wall of the Bowman's capsule. So small molecules, and if you look at the list there, we're talking about glucose, amino acids, and urea, importantly, because we want to get rid of that, all pass out into the filtrate. Large molecules like plasma proteins and red blood cells and white blood cells, they remain in the blood plasma. So that's why the Bowman's capsule is referred to as a molecular sieve. And in the keynotes you can see a nice little picture there illustrating we've got, if you think about our capillary wall, we've got a permeable capillary wall, so small gaps between the capillary cells, holes, pores in the basement membrane, and pores between the podocyte feet. So really it is a little sieve, like a molecular sieve. The pressure in the blood, because of the wide vessel in, narrow vessel out, means within the glomerulus capillaries there is a high hydrostatic pressure forcing fluid and small molecules across the capillary wall, basement membrane, and Bowman's capsule wall into the nephron, creating the filtrate. Okay, so let's have a little review. Okay, so I do like this picture. So this is a sort of microscope image showing all of those gaps and pores between the blood and the nephron filtrate. So you can imagine all the water, the glucose, the urea, the ions all being forced out of the blood plasma into the fluid that forms the filtrate. Okay, so stop here and have a little moment to reflect and ask yourself these 3 questions. What creates the high hydrostatic pressure in the capillaries of the glomerulus? Why is the Bowman's capsule known as a molecular sieve? And what molecules are forced out of the blood here? Pause. Okay, you should be back. Hydrostatic pressure, remember that was created because we had a wide vessel supplying, so an arteriole with a wide lumen supplying the blood to the glomerulus, and then a narrow lumen arteriole taking the blood away. So that's what created the high hydrostatic pressure. Why is the Bowman's capsule referred to as a molecular sieve? So pores, pores and pores. So we have a permeable capillary wall, so gaps between the cells of the capillary wall, which we can see clearly in here is 1 cell thick as well. Okay. We've got holes or gaps, pores in the basement membrane, and then we've got gaps between the podocyte feet of the Bowman's capsule wall, so gaps and pores. Okay. And what molecules are forced out here? Well, luckily, urea, because it's very small and we want to get rid of it from the body, it's toxic. But then, unfortunately, everything else about the same size as well is going to get lost into the filtrate. So that means we're forcing things out like glucose, amino acids, ions, and of course urea. They often like to work with you thinking about what's moving in application questions, as you'll see. So basically, everything other than proteins and large molecules gets forced out of the blood plasma at the Bowman's capsule. So that means the rest of the kidney really is about reabsorbing everything that the body needs back out of the filtrate so it doesn't end up in the urine. There are some conditions which have a damaging effect on the glomerulus. So if you think about someone with high blood pressure, an AOT question about the impact of high blood pressure on somebody and the glomerulus and Bowman's capsule, part of the nephron, talks about sometimes you might find blood or proteins in the urine of someone, and that would indicate that they might have high blood pressure. And that would be because high blood pressure would mean that this hydrostatic pressure was even higher than normal. That would damage the walls of the Bowman's capsule and the capillaries and result in larger molecules then making their way into the filtrate that shouldn't normally move into the filtrate. So that's an AO2 type question that you might see. I think there's one in one of the homeworks actually, which I'll make sure I send out. Okay, so now we've forced everything, water, ions, glucose, amino acids and urea, out of the blood into the filtrate of the kidney, of the nephron. We now need to reabsorb loads of it because we don't want to lose it in the urine. So we're moving into the second part of the nephron, which is the proximal convoluted tubule. Okay, so in your keynotes you move into the second part here. Okay, so here around 80% of the reabsorption of all the things that you don't want to lose in your blood, lose from your blood in your urine occurs. So that includes water, but all of the glucose, all of the amino acids that have been forced out under that high pressure in the Bowman's capsule are going to be reabsorbed back from the filtrate into the blood. And this is mainly because of the specialised adaptations of the proximal convoluted tubule cells. So this is a good one for essays because you already know all these adaptations, but it gives you a higher level context than talking about the small intestine and absorption of products of digestion if you go for the proximal convoluted tubule. So just have a little pause there and have a think about how you think from this picture this convoluted tubule cell is adapted for all this reabsorption. So I'm hoping straight away you've identified the microvilli, increasing the surface area of the cell surface membrane to increase the rate of diffusion, active transport, movement of molecules across that membrane. Remember, on the new spec you would need to talk about an increased surface area, would mean that there's more proteins, carrier proteins, channel proteins as well for the movement of molecules across that membrane. I'm hoping you've also noticed that we've got lots of mitochondria, so lots of mitochondria to produce the ATP required for active transport of molecules across that membrane. So in your keynotes, in bullet points here, you've got a nice little summary. So we've got our many microvilli, our many mitochondria. The other thing you couldn't see in the diagram is that there is a close association as well with capillaries. So the diffusion distance for movements of molecules is short, maintaining a large surface area for exchange. Slightly off spec here, okay. Larger molecules such as proteins with small molecular masses, the ones that had been forced out because they're just small enough, can be reabsorbed here, but they have to be reabsorbed using vesicles, which you can see quite a lot of as well in the picture, and that would be by endocytosis. Okay, so all of the glucose, all of the amino acids are reabsorbed by the proximal convoluted tubules. So straight away you should be thinking to yourself, then if it's all the glucose and all the amino acids, then they will only move by facilitated diffusion back into the blood while there's a concentration gradient. So to get all of them back, you will have to use active transport as well. Because of all the solutes moving back into the blood, the other thing that you'll notice here is that 80% of the water from the filtrate is reabsorbed straight away here at the proximal convoluted tubule. So that's happening because, obviously, as the solutes move by diffusion and active transport back into the blood, they're lowering the water potential of the blood, and water is following by osmosis. Okay, so back now into the PowerPoint for a quick check. Okay, so what happens in the proximal convoluted tubule? How are the cells adapted for this process? And why are they associated so closely with so many capillaries? Pause there. Okay, so what happens? Lots of reabsorption. That's what happens in the proximal convoluted tubule. How are the cells adapted? Well, we've got many microvilli to increase the surface area of the cell surface membrane. We've got many mitochondria for active transport of, to provide ATP for active transport. And as we said, there's a close association with capillaries. So why such a close association? Well, that will be to reduce the diffusion distance and increase the surface area for diffusion and active transport to happen. So for absorption. Okay, so we've gone through the proximal convoluted tubule, so now we're going to enter the loop of Henle. Okay, before we do, can I ask you to move into your question pack and just recap what we've done so far by answering question 1. So pause there, answer question 1 and then rejoin. Okay, so having a look at question 1, the first convoluted tubule is another name for the proximal convoluted tubule, so it's the first one. In the human nephron, sodium ions, glucose molecules and water molecules are reabsorbed into the blood plasma. Figure 3 illustrates how these substances are reabsorbed. Explain the large number of organelles labelled X in these cells. Give 2 differences between the process by which water enters the capillary from the epithelial cell and that by glucose and sodium leave the epithelial cell, and explain the difference in the structures labelled Y on the epithelial cell. Okay, so many mitochondria again. So we've got mitochondria, the site of respiration, produce ATP, needed for active transport. This is old spec, so you might just need a little bit more detail. You'll definitely need the site of production of ATP, and that the ATP is required for active transport. For the next one, this is very new specky. So water leaves the epithelial cell passively, doesn't require ATP. It's down a water potential gradient. Now, slightly different on the new spec, it says without the aid of carrier proteins or transport proteins, but of course we have aquaporins now on the spec. So you would say with aquaporins being a specific type of protein, whereas glucose and sodium would use a different carrier protein type. For sodium and glucose, the idea that it would require active transport, so there would be some ATP required, would be down a concentration gradient, but then obviously would be up a concentration gradient as well. So the final one then is the microvilli to increase surface area for absorption. Okay, so make sure you've checked all those off. Okay, so entering now the loop of Henle. Okay, so for the loop of Henle, normally I would draw on this diagram on the board. So what I'm going to do is reposition myself so I can draw on the desk in front of you. Okay, so it's a kind of bog standard diagram this. So this is my loop of Henle. So this is the descending limb and this is the ascending limb. Okay, and in terms of the position in the kidney that you find the loop of Henle, so the proximal convoluted tubule would be in, the Bowman's capsule and the proximal convoluted tubule would be in the cortex, but then as you go down the descending limb of the nephron, it comes deep into the medulla tissue. So this is the medulla tissue fluid. There's lots of capillaries I haven't drawn on as it just makes the diagram look really messy. So anything that moves into the tissue fluid at the medulla, there are lots of capillaries in there that would absorb those molecules into the blood. So this story is one of countercurrent. So just like when we were looking at the gills, we need to think about why this is the most adapted it can be to ensure that most water is reabsorbed here. So if there's water left in this filtrate, the majority of it is getting pulled back, or a large proportion of it is getting pulled back into the medulla and then into the blood. So the story actually starts over here at the top of the ascending limb. Okay, so at the top of the ascending limb, what happens is the cells actively transport sodium and chloride ions out of the filtrate and back into the blood via the medulla tissue fluid. Okay, so that's happening all the time, and that lowers the water potential here in the medulla. So we've got a lower water potential in the medulla tissue because we are pumping out sodium and chloride ions. Okay, but this limb, this ascending limb, is actually impermeable to water. The descending limb wall isn't though. So as the water potential is lowered in the medulla by pumping out these ions, then water is drawn out of the filtrate, but it can only move across the permeable descending limb. So water moves out by osmosis down a water potential gradient, but only here in the descending limb. Okay, so if you imagine that's happening like that, all the way down this beginning bit of the descending limb, water's going to keep moving out down a water potential gradient because the presence of those ions is lowering the water potential of the medulla compared to the filtrate. Now, what I want you to think about next is what's happening to this filtrate concentration. Okay, so, no ions can leave. But if I measured the sodium ion concentration at the top here, and then measured it again here in the filtrate, I would find that its concentration had gone up. Now, the ions haven't moved anywhere, but water has been lost. So everything is becoming more concentrated in the filtrate here. Okay, now that could cause us a bit of a problem if you think about water potential. So now this water potential here is becoming lower, so it's lowering. So what we really need is a very, very, very low water potential at the bottom of this limb here to make sure that we can still draw water out by the time we come to the bottom. Okay, now the descending limb, although permeable to water, what's really important, I suppose, to tell you to understand why that concentration of the filtrate is going up, is that it's impermeable to ions. So they have to stay inside the descending limb. Water's moving out. Therefore, it's becoming more concentrated inside the filtrate. Okay, so why does water keep moving out then? Well, we have to start to think about what's happening here as we turn the corner at the apex. Here, the walls of the ascending limb become permeable to ions. Now remember, by the time you turn this corner here, the concentration of sodium and chloride ions are going to be really, really, really high compared to the medulla. So there's a rapid diffusion of sodium and chloride ions here. Now that's really important because that means lots of the ions move out here, which means that water is still moving out by osmosis just before the bend, because here is a very, very, very low water potential. So even though this one's not quite as low, quite as high as it was, the water potential as it was at the top, because water's been moving out, we've still got a higher water potential in the filtrate than the medulla tissue. Okay, so those ions move out of the ascending limb, first of all by diffusion, and that happens really rapidly, and then by active transport when you get to the top. This is really important because by doing that, we've always got a water potential gradient along the entire descending limb to draw water out into the medulla tissue. Okay, that's really important, and it's because there is a counter current. So by that we mean a counter current of filtrate. So this filtrate is flowing in this direction down and in this direction up the limb. Okay, so we need to recap that. It usually takes a couple of times to go in. Okay, so the first thing I always talk about when I answer these exam questions, describing how the loop of Henle and the countercurrent system within the loop of Henle ensures that lots of water is reabsorbed from the filtrate back into the blood. I always start here. So I start with my first mark point. I say ions are actively transported out of the filtrate into the medullary tissue. At the top, or towards the top of the ascending limb of the loop of Henle. Okay. And the second thing I talk about is that what results from that. This lowers the water potential in the medulla. Okay, so the movement of those ions out lowers the water potential in the medulla. Now, although not necessarily on the mark scheme, it's important to note now that this wall, our ascending limb, is not permeable to water. So water cannot follow by osmosis from the ascending limb. The descending limb walls, however, they are permeable to water. And the other thing to note is, although the descending limb walls are permeable to water, ions cannot move through them, so they cannot move out. So they're important rules to remember for this to work. Okay, so ions are actively transported out at the top of the ascending limb. This lowers the water potential in the medullary tissue, so that the water potential in the medulla is lower than the filtrate. Third mark point, therefore, water moves from the filtrate to the medulla by osmosis. Now, quite often that might be all there is on the mark scheme if it's a short mark scheme. But the really difficult bit of this is understanding why this countercurrent ensures that water potential gradient I've just described is always present. And the only way to understand that is to think about the filtrate. So by the time you get to the bottom of the descending limb, the filtrate becomes very concentrated. In other words, its water potential is lower than at the top of the descending limb. We've lost lots and lots of water. So if water is going to continue to move out by osmosis, the medullary tissue here at the apex, at the base of the loop, would have to be very, very salty, very, very concentrated, so that it's still got a lower water potential than the filtrate, and water still moves out by osmosis. Okay, and it's because this filtrate is passing in a countercurrent that's created, because now the filtrate, being really concentrated, flows in the opposite direction up the medulla, which means that now the wall becomes permeable to ions but impermeable to water. Ions diffuse rapidly out of the filtrate causing the medulla tissue at the apex to have an even lower, so I usually got even lower water potential. An even lower water potential at the apex. Okay, and that means that water will still move out here. Okay, so it's called a countercurrent multiplier, and it ensures that we reabsorb the maximum amount of water we possibly can from the filtrate in the loop of Henle. Okay, so if you turn back into your keynotes here, you can see that you've got that described, hopefully, in bullet points that match up with this diagram here. So, number 1, this is the active transport of sodium and chloride ions out at the top of the ascending limb. This lowers the water potential in the medulla, but remember the ascending limb going up is impermeable to water, so water can't move out of the ascending limb, but the descending limb is permeable to water, and as the water potential is lower in the medulla, water leaves the filtrate in the descending limb by osmosis. Once it gets into the tissue fluid of the medulla, it then just moves back into the blood and water potential gradient. Remember, ions cannot leave the descending limb. That means by the time we get to the bottom of the descending limb, the filtrate ion concentration is much higher. That means at the apex here, you've got the highest ion concentration. So that means as soon as you turn the corner, sodium and chloride ions diffuse out of the ascending limb down a steep concentration gradient. This lowers the water potential here in the medulla at the apex even more, and means that water continues to follow, moving out of the descending limb by osmosis. So here it says the countercurrent multiplier at the loop of Henle results in a maximum water absorption. And the things that you would talk about when answering that are the characteristics that ensure that maximum reabsorption. So the descending limb, remember, is permeable to water but not ions. The ascending is permeable to ions but not water. Active transport of ions at the top the ascending loop, when the ion concentration is dropping. So if you think about here, ions are moving out by diffusion, and the concentration of ions is starting to get really low. So we now need to start pumping them out against a concentration gradient. That ensures water is always moving out of the descending limb. So we've got concentration a water potential gradient along the whole of the limb. Bit of an application question that sometimes comes up is about adaptations of organisms. So if you have a look at an aquatic species, a beaver, compared to a human, compared to a kangaroo rat, the longer your loop of Henle, the larger the surface area, if you like, for reabsorption for these things to happen. So you would reabsorb more water the longer your loop of Henle. Okay, so a quick check, and then question 2, and then we're going to pause before we go on to the next section. So what does the countercurrent multiplier mean? What does it mean? What conditions are established in the medulla? How do these conditions establish, and how does this affect water reabsorption? So pause here and have a go. Okay, so you should be back with me. So the countercurrent multiplier, if I was asked that in an exam, what is it? It's the flow of the filtrate down the descending limb and up the ascending limb, and then I will give the features of the limbs. So I'd say that the descending limb is permeable to water but not ions. did I say the right way around then? The descending limb is permeable to water, not ions. The ascending limb is permeable to ions, not water. Therefore, having the filtrate in the flow in opposite directions, down and then up the limb, ensures that the water potential gradient is maintained across the length of the loop of Henle. So what conditions are established in the medulla? Well, the medulla has always got a lower water potential than the surrounding filtrate. So it's got a higher solute concentration. How is this established? At the apex, through the diffusion of ions out, but later on, when the ion concentration is lower, through the active transport of ions out of the filtrate into the medulla. How does that affect water reabsorption? As we said before, it ensures there is always a lower water potential in the medulla than the filtrate of the descending limb. Therefore, water moves out by osmosis all the way down the descending limb. So you'll get maximum water reabsorption. Okay, so if you move now into question 2 in your question pack and give this one a go. So in a mammal, urea is removed from the blood by the kidneys and concentrated in the filtrate. Describe how urea is removed from the blood and then explain how urea is concentrated in the filtrate. So this goes back to our Bowman's capsule, and this one is what we've just done in the loop of Henle. So if you think about urea, it's something that you wouldn't reabsorb, and it becomes concentrated when water is removed from the filtrate. So it's a backwards way of asking, how is water reabsorbed? Explain how water is reabsorbed from the filtrate. So pause there and give it a go. Okay, so answer to the part AI. High hydrostatic pressure in the capillaries of the glomerulus forces urea out of the blood across the permeable capillary wall and through the pores of the Bowman's capsule. Because it is small, and this is called ultrafiltration. That was my answer. My mark points would be for the high hydrostatic pressure in the glomerulus forcing urea out of the blood. 1 mark for the permeable capillary walls and pores in the Bowman's capsule, and an extra mark is for it's called ultrafiltration. What about this concentration? Well, it was only a short answer, so it is just the easy sort of description, hopefully. Oops, sorry. So sodium and chloride ions are actively transported out of the top of the ascending limb into the tissue fluid of the medulla. This lowers the water potential of the medulla, and water leaves the filtrate in the descending limb by osmosis. And that would explain why we're concentrating the urea. Four marks. I believe in the mark scheme it's got sodium chloride ions are either actively transported or diffused. On the new spec they might want you to have actively transported at the top, but sodium and chloride ions are diffused out at the bottom of the ascending limb, so you probably need that for the new spec. Both of those would lower the water potential in the medulla. You might then get an extra mark point for saying that diffusion at first and then active transport ensures there is always a lower water potential in the medulla than the filtrate, so that water leaves the descending limb by osmosis. So descending limb and osmosis will be separate mark points on this old spec. So it was tick for sodium chloride ions actively transported. Where it was happening didn't get you a mark, it was for lowering the water potential, and then water leaving the descending limb by osmosis. But as you can see, you probably need more detail now on a new spec question. Okay, so this had a very much new spec feel to it. real sort of application question. The diagram shows one way in which a person who had kidney disease can have the condition managed. In the process, a fluid is put in the abdominal cavity. Exchange of materials takes place across the membrane that surrounds the abdominal cavity. This removes waste products from the blood. After 5 hours, the fluid is drained out of the cavity and discarded. The cavity is then filled with fresh fluid. So the table shows the concentrates of solutes in the fresh fluid. So we've got very high sodium concentration, chloride, calcium, is very low, loads of glucose, no urea at all. Okay. By what process does urea enter the abdominal cavity from the fluid in the abdominal cavity from the blood? Well, we know in the blood it would have a higher concentration than 0, so we know it's going to be diffusion. That's your first mark point, simple diffusion. Explain why the fluid has to be changed every 5 hours. So again, this is just application of movement of molecules, because if you didn't, there wouldn't be a concentration gradient, it would reach equilibrium. The idea that you need to maintain concentration gradients by exchanging the fluid, so the fluid always had higher concentrations or lower concentrations of the things you wanted to exchange. It says the fluid of the composition shown in the table was used instead of distilled water. Explain why. Well, what you want to happen is for urea to move from the blood into the fluid, but what you don't want to happen is things like glucose and ions that you need in your blood would also move. So you can see they're in quite a high concentration in the fluid. Okay, so what they're looking for is this idea that the ions, the glucose, the amino acids would diffuse out into the bag if you use distilled water, because they would be moving down a concentration gradient, so into the distilled water, because of course they would be higher in the blood than the distilled water. Okay, and that could then cause a deficiency in those substances. So that was one reason. The other way you could have answered it was to talk about the water potential. So if you use the distilled water, okay, then the water potential would be higher than the surrounding tissues, and so water would move into the tissues by osmosis in the abdominal cavity, cause the cells to be damaged or burst. So that was also an acceptable answer. So either way, just applying your knowledge of movement of molecules across membranes, which is a lot of what they do in the kidney when they're asking you to do AO2. Okay, so we're going to stop there, and I'll ask you to review what we've done so far. It's quite tricky, that loop of Henle, and then we're going to start to pick up on the distal convoluted tubule in the next video. Okay, thank you.